Hi, this is Conor Houghton, and I'm going to talk about the Hodgkin-Huxley equation. So we've been talking already about the dynamics that governs the voltage of a neuron. The equation we got to looks something like this. We have Cm dV dt is equal to GL EL minus V plus inputs. And so the idea here is that the cell has an inside and an outside, as we discussed, and the inside has a voltage V relative to the outside, which we're taking to be 0. And the change in the voltage, well, we have this differential equation for that, where basically V chases EL. This is the conductance, that's the propensity of the membrane to conduct. This is the capacitance. When we divided one by the other, we got the membrane time constant. We won't do that here, it's not convenient. The point was that this GL was the fixed conductance. It was a conductance through the membrane, 1 over the resistance, that didn't change as the voltage changed. And what we said at the time was that was an approximation, that in fact the membrane contains channels that change their conductance. In other words, there are holes, as it were, in the membrane. And they can open and close, changing the conductance of the membrane as the voltage changes. And the effect of that is that it allows the cell to spike. So if you remember, the neuron has an axon, and spikes of voltage travel down the axon, and that's what carries the signal from neuron to neuron. That spiking depends on the fact that there are these channels, that there are the conductance GL, or the conductance G rather, changes as the voltage changes, that the conductance has some dynamics. That's all been left out here, and if you remember, this is an integration fire equation. The and fire bit says that whenever the voltage reaches some threshold, we reset the voltage and we say, well, there was a spike, and in our network model, if we have a network model, we send the fact that there's a spike to all the other neurons in the network. But we might want to do something more. We might be interested in the dynamics of the cell. We might want to understand or model the production of the spikes. That equation was discovered by Hodgkin and Huxley through a series of rather heroic experiments, certainly very pioneering experiments, on the giant axon of a squid. Their equation, as we'll see, is phrased largely in terms of the transport of sodium and potassium. That's not necessarily true of all the cells that people study these days. And the simple picture they have, where there's one type of gate for sodium and another gated channel for sodium and another type of gated channel for potassium, is very much an approximation, or at the very least a description valid for the particular axon they were dealing with. But telling the story that they tell is enough to help us to understand how spikes could be formed by the dynamics of these gates. I have a picture of one of these gates here. So this is a potassium gate. As you can see, it's this very, very complicated molecule, and it has a few sort of interesting properties. The first, of course, is that it's ion selective. Only this is the potassium traveling through in the braided middle, and only potassiums can travel through, to a very good approximation. But the second point is that this gate opens and closes. And so here it is illustrating the open state. It's allowing the potassium to go through. The idea is that there's also a closed state which would prevent the passage of the potassium. And what we're going to model is the transition between this closed and open state. I have a little picture here. This is from the sort of top. That's the hole that goes through the membrane. So we have a picture here from the side. It's not such a good picture, in fact. Uniquely of these pictures, I think I drew it myself years ago. This is the potassium gate that we're looking at here. It's from the side. You can see that it's in a closed state there, blocking the passage of the potassium. And here's the open state. And we're interested in the fraction of the gates that are open and closed, or the transition or the probability of a given gate being open or closed. And that will give us the voltage dependent, or the dynamic, the time dependent conductance of the potassium channel, and that will be important for the dynamics that produces the spike. Now, Hodgkin and Huxley didn't know about this picture. I mean, they presumably believed that it would at some point in the future be possible to produce a picture of the molecule that is the gated channel. They lived at a time when people were using crystallography to discover the structure of large biological molecules, but none this complicated or this hard to crystallize, etc., etc. And so they weren't working from this picture. They were working phenomenologically. They had an idea, which was that there was something that opened and closed, and they were trying to come up with equations for it. And they were expecting or hoping for equations of this form here, where you have some time constant, you have something that's changing with time, and that's going to be equal to some asymptotic value minus L. In other words, L will head towards L infinity, and it will do that with a speed tau L, and though they expected that this would depend on the voltage, and this would depend on the voltage here as well, tau L of V. But what they discovered was that the convenient thing was not to have an L which described the conductance of this whole molecule. They realized that for the gates they were dealing with, they were better off dealing with something whose power described the conductance of that whole molecule. And I should have really opened with this and said we can see that this molecule has a C4 symmetry. It's obviously made of 4 subgates. And so what we're going to do is to write down a dynamical equation for the subgate. So what I'm trying to say is the following. We can imagine the gate, this is the gate here. It has, as it were, 4 kind of blockers, and each of those blockers corresponds to a subgate. And what we're going to do is write down a dynamical equation for the probability of one of those blockers being open or closed. So if you imagine for a given gate, if the probability of one of the blockers, let's call it n, that's the probability that one of the blockers being open. Well then, if the 4 blockers, as it were independent, if the 4 subgates are independent, then the probability of all 4 being open is n to the 4th. That's not, again, as with all these biological things we talk about, that's not necessarily such a good approximation. We know now that there's complicated transition diagrams for different states. In this case, the state when there's 3 blockers open or 4 blockers open, and I myself have worked on a particular gate, that's a research in sodium gate, where it is important that there is this complicated set of dependencies and that the individual subgates are not independent. But a lot of the dynamics, and certainly the dynamics we're interested here, doesn't depend, or is well approximated by thinking of the 4 subgates as open. So what I'm trying to say is that what we're going to do is look at the equation for the probability of one of the subgates being open, knowing that the probability of all 4 being opened is then, or approximating the probability of all 4 being opened with n to the power of 4. Something that we can sort of see in the molecule, but something that Hodgkin and Huxley, as it were, discovered phenomenologically. They just found that it was easier to get their equations to work if they worked like that. You can also somehow see why it would be the case biologically. I mean, if this is, the axis is funny here. If this is n here, this is n, and this is n, and so n is a straight line. That's what n looks like. Whereas n to the 4th, of course, looks like, sorry, we're going from 0 to 1. N to the 4th would look, even sharper like that. And so by going to the n to the 4th, the biology has chosen a system which will accentuate or make crisper the transition between 0 and 1. Okay, so to back up a little bit, what's the plot? The plot is that we were taking the original equation that we argued for before, but before we talked about how there are currents, and the currents have a reversal potential, and so they give this leak current that looks like that. But we're going to add more currents. There's going to be a sodium current that has a different reversal potential. That actually is about 20 millivolts, and we're going to add a potassium channel, and that has its own reversal potential. That reversal potential is about minus 80 millivolts. And then, as before, there's inputs. And so the idea is that before we just had a single channel, the leak channel. Its conductance was constant as the voltage changed. as time passes. But we have 2 more channels now, and they're ion selective channels. In other words, this is a channel specifically for sodiums, and so the relevant reversible potential is just the sodium reversible potential. And then there's a potassium channel, and again, this is only for potassiums, and so the relevant reversible potential is potassium. And the cool thing is that these give quite different values. This is plus 20 millivolts. In other words, the sodiums are trying to get in, and this is minus 80 millivolts. The sodiums are trying to get out. So, well, what next? Again, just the usual health warning. We're just going to pretend there's one type of sodium channel. In fact, there generally is not that many types of sodium channels. In a given cell will usually only have 1 or at most 2. And then we're pretending there's only 1 potassium channel, when in fact cells often have quite a few potassium channels. We've left off the more complicated chlorine channels, are quite rare, but we've also left off the calcium channels, and they can be quite important, particularly because calcium also plays a role in plasticity, and because there are complicated channels that we're not going to talk about at all, where you have an interaction between the potassium channel and the calcium channel. Either way, this is what we're going to do. Let's concentrate initially on the potassium channel. So the conductance for the potassium channel, we're going to let that equal to some maximum value by some probability, and the probability we're going to write as before is n to the 4th. So this is, you can think of this as the fraction of open channels, or the probability that a given channel is open. And so this is the maximum value, the constant bit, and this is the bit that's a number between 0 and 1, whose dynamics we're going to study. And again, we now just think about what the equation might look like. And one way of thinking about the equation is in terms of transition probabilities. So the increase in n is going to be equal to alpha times the number of channels that are opening. So these are the channels that are closed that are now open. That increases n, but it's also minus the probability of a channel closing. So these alpha and beta are transition probabilities. N is a number between 0 and 1, and it's just a probability, so it's a dimensionless number. This is per time then, dn dt. And so this is a rate, and this is the rate. So this is the rate that closed channels become open. This is the rate that open channels become closed. The number of open channels, the fraction of channels that are open, is n, and so beta times n is the rate at which the number of, in terms of density, the fraction of open channels becoming closed. This is the fraction of closed channels, and so this is the fraction of closed channels that are becoming open. And that's increasing n, that's decreasing n. We can then rewrite this, so it's dn dt is equal to alpha minus alpha n minus beta n, which is equal to alpha minus alpha plus beta times n. And so you can see what we're probably going to do, which is divide across by alpha plus beta. So we get 1 over alpha plus beta dn dt is equal to alpha over alpha plus beta minus n. Okay, and so that is a rate, because it's 1 over, that's a time scale. 1 over a rate is a time scale, and so 1 over alpha plus beta is a time scale, so we're going to rename that tau n, and alpha over alpha plus beta is dimensionless. We're going to call that n infinity. So you can see, I start off by describing this in a simple way in terms of transition probabilities, but we can also write it in a way that looks a lot like the equations we've studied before. So I'll write that again at the top here. So we have we have tau n dn dt is equal to n infinity minus n. And in our usual interpretation, n is heading towards n infinity. That's what we call it infinity. It's where n would be after an infinite amount of time has passed, and tau n is just a time scale. But the difference is that here we believe that tau n and n infinity depend on the voltage. So tau n and n infinity depend on the voltage. They depend on time as well, but only through the dependence of the voltage. Whereas dn dt has sort of time dynamics. It will change with time because it's trying to follow tau infinity, the thing that depends on the voltage, and it does it with the timescale tau n, and that also depends on the voltage. I'm just going to show you pictures of what these look like. So what Hodgkin and Huxley did was they played around with different sort of functional forms of n infinity and tau n until they got something that kind of gave the right behavior. And those equations, weren't derived by them. They were discovered phenomenologically, and we still don't really have a derivation as such for them. So here, where is n? This is n here. It's a bit hard to see, but basically this is 1, this is 0, this is minus 100 millivolts, this is 20 millivolts. You can see it really starts kicking off around minus 60 millivolts. So that's a picture of what n infinity does. It shows you that for low voltage, n infinity is near 0, and n to the fourth is even more near 0. So you can see the advantage that biology has discovered by having a fourth power, whereas near when the voltage is high, it's pretty close to 1. And that's close enough to 1 that by the time you take the fourth power, it's still close to 1. So as the voltage increases, n infinity is going to increase towards 1. And so that means n heads towards n infinity, and it also increases towards 1. And what that means is that the conductance, the potassium conductance, is increasing. And what is that time scale? Well, I have a picture of that here as well. And you can see that the time scale for tau n, which is this line here, things start happening a bit quicker as the voltage gets higher. But 5 milliseconds, which is this value here, is a sort of indicative time scale. So you can see that the time scale, it varies between 2.5, or maybe that's even 1.5 and 5 milliseconds, and it does get faster as the voltage gets higher. So that's the dynamics of potassium. To tell the full story, though, we really need the sodium dynamics. So we got the n was coming from, again to remind you, Gk is equal to the maximum value by n to the fourth. What we want now is to look at the conductance for the sodium. And the sodium conductance is more complicated. We have Gna is equal to the maximum value, and instead of the n to the fourth, we have m cubed h. In other words, there's two types of gates. There's the m gate, which people might sometimes call the opening gate, and there's the h gate, which is the closing gate. And where the potassium has a fourfold symmetry, so it's invariant as you rotate it through pi over 2, the sodium has a threefold symmetry, so it's invariant as you rotate it through 2 pi over 3, and so it has the third power here. But it also has this h gate, and both of them satisfy the same dynamics, so the same sort of discussion we had before leads to tau m dm dt is equal to m infinity minus m, and tau h dh dt is equal to h infinity minus h. So they both satisfy the same equation, but the difference is that the h infinity goes from high to low, whereas the m infinity, like the n infinity, goes from low to high. Let me first of all sort of say kind of in words the plot. So the idea is that we have the gate here. This is supposed to be a picture of the sodium gate. And it has these two kind of blockers. This is this sort of M blocker, the one before, the opening gate, and this is H, the other blocker. And so when the voltage is very low, it looks like this. The H gate is open, but the M gate is closed. Then as the voltage gets higher, so this is in the sort of closed 1 state, people sometimes call it. As the voltage gets higher, the M gate opens, but the H gate also remains open. And so this is the open state, and this is when the sodium can pass through the channel. But as a little more time passes, so this obviously the time constant here is a bit longer, while the M is remaining open, the H is closing, and now we're in the other closed state. And I have a little picture of that here as well, which again is just hand drawn by me. So that's this picture here. This is the closed 1 state, where it's the this gate here, the one that looks a lot like the potassium gate that's closed. Then that opens, but a little bit more time passes, the H gate closes. And that sequential behavior, the M gate opening first and then the H gate closing a little bit afterwards, well, that sequence is determined by the time constant. Basically, the time constant for the M gate is very small, the time constant for the H gate is somewhat greater. So here we go. So this here, down here, these are the time constants. That very low time constant there is the time constant associated with tau m. And you can see it's rarely, it does slow down a tiny bit as you go, but it rarely is much different from like half a millisecond or whatever. And people sometimes, in approximating logic and Huxley, just pretend that m is fixed always at its m infinity value, because really it has a very, very small time constant. The h, though, has a much slower time constant. In fact, around the threshold, which is when everything is kicking off, you can see that the tau h time constant is right up at 7.5 milliseconds. So the h stuff happens slower than the m stuff. In fact, the m stuff is the very fastest thing that happens, and then you get the m stuff, and the m stuff is the fastest, then the n stuff, which is the potassium, and then the h stuff, which is the closing gate. And conversely, this is a picture of the infinity values. So here, that's the h gate. You can see it's completely the other way around to the m gate, and the m gate is here, and the n gate we drew before. So these are the opening gates. These are the gates where they get up to 1 as V heads towards 20 millivolts. So the higher the voltage, the more open the sodium gate is, and the more open the potassium gate is, as far as M and N are concerned. But as far as H is concerned, which closes the sodium gate, as V increases, it goes very much down to 0. You can see that you're already at minus 20 millivolts. It's very much exactly 0. And so there'll be nothing passing through the sodium gate if H approaches its asymptotic value, H infinity. But we know, of course, that it approaches its asymptotic value reasonably slowly at the timescale of a spike because its timescale is a bit longer. And so that's really all we need. That's enough to give us the picture of how the spike occurs. So the way that the spike occurs is, here's my voltage, V. This is minus 80 millivolts. Not much is happening here. This is minus 50 millivolts, where everything starts happening. This is 20 millivolts right at the top. So we're wandering around here, not much is happening. Now, suddenly, we're getting to the minus 50 millivolt point. At that point, the M gate is starting to open. And so what does that do? It means that sodium starts rushing into the cell. So at this point here, sodium is rushing into the cell, because at this point here, the M gates are opening. Okay, now that of course now pushes the voltage upwards, because as the sodium comes in, sodium is a positive ion, it increases the voltage. And so quite rapidly, this isn't really the scale, the 20 millivolts should be right up here, the voltage starts heading up towards 20 millivolts, which is the reversal potential of sodium. Now, you'd think that that's actually just going to keep going, and eventually the sodium will all end up in the cell until the inside of the cell has the same concentration as the outside. That'll be no fun, and that's not what happens. Instead, as it goes up, what you start to see happening is that the H gate starts closing. So the H gate starts cutting off the inflow of potassium. So at this point here, H closes and stops the potassium, stops the sodium coming in. And so this influx of sodium that was causing the upswing in the voltage is cut off by the H gate closing. At the same time, the N gate opens. And so what that means is potassium starts leaving the cell. And of course, as potassium leaves the cell, so there's no more H coming in. The H really does come right down to 0, as you can see. So as the H approaches H infinity, there really is no more sodium coming into the cell, but instead potassium is rushing out of the cell, and that pushes the voltage back down. And that's the story of a spike. You can see it's this sort of cunning sequence of events where you have the very fast opening of the M gate. The sodium rushes in, that pushes up the volume, the voltage. Then the H gate, which has a slower time scale, but it starts causing the closing of the gates, and well, what happens then? The potassium stops coming in, but at the same time now the sodium gates are opening and the sodium starts leaving, and that pushes us right back here. Now, you of course, you'll notice that when it's back here, the cell hasn't quite, the voltage has returned to where it was before, or even further down than it used to be, but everything hasn't returned to normal because of now, of course, the sodium gates are going to be closed as in the closed 2 state with the closed H and the open N, and the amount of sodium in the cell has decreased slightly, the amount of potassium in the cell has increased slightly. So it's not like this and this leave the cell in the same state. However, once it's down here, things could have turned back around. So, once we're down here, you can see that, again, the M gates will shut rather fast. Sorry, where's the M gates? The M gates will shut rather fast. At the same time, the H gates will start opening again. The potassium will have closed by now because we're right down here. So potassium, of course, is back here. And over, a timescale of a few milliseconds, potassium will close again. And so there will be a kind of return to normal. And the pumps get working away so that the potassium that left will get pumped back in, or not necessarily the same ions, but the same amount of ions, and the sodiums that came in will be pumped back out. But of course, because the potassium gates will remain open for a few milliseconds, that's what gives the cell its refractory period. And it's crucial, of course, that the sodium gates, M gates, close before the H gates open. That prevents a resurgence of the sodium into the cell. But there are other cells where those things don't happen so much. So there are other cells where you have a smaller times constant in the potassium for low voltages, for example, which will give a shorter refractory period, or you could have the sequence of the opening and closing sequence for the sodium's slightly different, and that would allow a resurgence in the sodium, for example. So there's other stuff that can happen, but this is a standard sort of story. And so again, just to recap before we finish, we have in the Hodgkin-Huxley equation, so we have Cm dV dt, that's the capacitance as before. We have the stuff that doesn't depend on voltage, which is fine, and then we have two other currents, the potassium current and the sodium current. I keep pointing out that the potassium current actually often comes in a few varieties, so there'll be a few of these terms here, and they'll have different time constants and so on, allowing for more complicated dynamics. What they call fast sodium, which will help with the spikes, and then slow sodium, which causes the sort of spiking refractoriness that we talked, the spiking adaptation that we talked about before. There's other channels to do with calcium and so on, and then finally there might be inputs from the dendrites or in an experiment from an electrode. And all of these terms, well, they're made up of subgates. Subgates all satisfy equations. Well, subgates we looked at all satisfy equations that look like this. In particular, the sodium has 3 M gates and a H gate. This has 4 N gates, and the particular sequencing of their time constants and their asymptotic values allows, first of all, sodium to rush into the cell. The sodium gets cut off, the potassium rushes out of the cell, it forces the voltage back down, and then there's a period where everything gets reset. And that is the Hodgkin-Huxley equation and the story of where spikes come from.